These stem cells produce two distinct types of immature lymphocytes, B and T lymphocytes. T lymphocytes, or T cells, are the main players in the cell-mediated response. Although stem cells are produced in the bone marrow, T cells must mature and differentiate in the thymus. In the thymus, T cells develop their ability to recognize the enemy, an antigen. The ability to recognize a foreign antigen depends on unique receptor proteins on the cell membranes of the T cells. The receptor proteins of each T cell are tailored to respond to only one antigen. During the cell-mediated response, a macrophage or other antigen-presenting cell is the first to detect an invader. The macrophage recognizes the antigen as foreign and engulfs it. Lysosomes break down the antigen. Antigenic fragments are displayed on the macrophage cell membrane bound to MHC proteins. Just as the sight of a red bandana triggers an attack by blue team members, the combination of an antigen and an MHC protein on a macrophage cell membrane activates T cells. Activated T cells divide rapidly and multiply into an army of specialized T cells. Some destroy antigen-bearing cells, while others help regulate the immune response. Each type of T cell is programmed to recognize the invader that triggered the original macrophage attack. The first is the cytotoxic or killer T cells that carry out the cell-mediated response. Cytotoxic T cells travel to the infected area and attack cells whose membranes contain the antigens that triggered their activation. They destroy the target by releasing toxic chemicals. Another is the memory T cells, which remain in reserve. If the same antigen appears again, memory cells immediately develop into cytotoxic T cells, providing a speedy and effective defense. Helper T cells are the commanders of your defensive forces. They integrate elements of the specific and nonspecific defenses and coordinate cell-mediated and antibody-mediated responses. Helper T cells also produce chemicals that stimulate the division and differentiation of T cells and B cells. And lastly, suppressor T cells inhibit all aspects of the immune response so that it ends when the infection has been controlled. The second specific defense mechanism is the antibody-mediated response. B lymphocytes, or B cells, are also produced in your bone marrow. Activated B cells divide to produce two types of defensive cells, plasma cells and memory B cells. As with T cells, each B cell is programmed to target only one specific antigen. Helper T cells also play an important role in the antibody-mediated response. They secrete chemicals that activate B cells and promote their differentiation into plasma cells. Plasma cells are the main players in the antibody-mediated response. When plasma cells mature, they release a flood of antibodies. Antibodies are powerful chemical weapons. They combine with the target antigens to form antigen-antibody complexes. The presence of antigen-antibody complexes acts like an ignition switch, turning on several mechanisms to inactivate or destroy the invader. Antibodies themselves may disarm the invader by blocking the attachment sites on a virus, or they can inactivate bacterial toxins. One method is to precipitate the toxic molecules by clumping them together, creating complexes too large to remain dissolved in body fluids. They can also agglutinate invading cells. These large clumps of cells are easier for phagocytes to locate and capture. Another mode of antibody action is to activate nonspecific defenses such as the complement system or the inflammatory response. Some of the daughter cells produced by divisions of activated B cells do not differentiate into plasma cells, but instead become memory B cells. Memory B cells become part of the body's defensive reserves. Some can survive for 20 years or more. If a memory B cell meets the same type of antigen at a later date, it differentiates into a plasma cell and begins secreting antibodies almost immediately. After viewing this material on the immune response, you can now understand the various mechanisms that your body uses to prevent the spread of disease organisms. Just as the two paintball teams identify invaders by their bandana colors, our body cells also have a complex surface marking system. Our white blood cells use this system to fight off foreign invaders and keep us healthy. Hi, I'm Susan Savoy, and welcome to the video tutor discussion of the respiratory system. After this man has been jogging for a while, he'll begin to feel his heart beating faster and faster, and he'll have to take deeper breaths. His lungs will expand as he takes in air. Let's follow the pathway an air molecule travels to enter the lungs.
Air enters through the nasal cavity, travels through the pharynx and glottis, along the trachea, and into the bronchi. The bronchi branch into more narrow passageways.
The individual airways may branch more than 23 times before reaching the respiratory surfaces of the lungs. These surfaces are divided into millions of microscopic air sacs called alveoli. Molecules travel along gradients from a region of high concentration to a region of lower concentration. Air under pressure acts the same way.
It flows from an area of higher pressure
to an area of lower pressure. Air moves from the atmosphere to the lungs in response to pressure gradients.
The lungs are held against the inner surface of the chest wall by a fluid bond. As a result, when the thoracic cavity expands, the lungs expand.
As the lungs expand, the air pressure inside the lungs also drops. The pressure differential between the lungs and the atmosphere causes air to flow into the lungs. When you exhale, the process reverses. The relaxed diaphragm bulges into the thoracic cavity and decreases its volume. Air pressure rises until it exceeds atmospheric air pressure and air flows out of the lungs. Exhalation slows and finally stops as air pressure in the lungs drops to match atmospheric pressure. The direction and rate of gas diffusion depends on the concentration gradient of the gases at the exchange site. In the alveoli, oxygen concentrations are high compared to body fluids.
Oxygen diffuses from the air in the alveoli into the capillaries. As oxygen diffuses across the respiratory membrane,
It's carried away via the circulatory system. On the other hand, the concentration of carbon dioxide is high in body fluids and relatively low in the alveoli. Carbon dioxide diffuses in the opposite direction, out of the blood and into the alveolar air. But the air we breathe isn't a single gas. It's a mixture of oxygen, nitrogen, water vapor, carbon dioxide, and several other gases. This bar chart shows the percentages of various gases in the air. Nitrogen and oxygen are the most abundant. They make up more than 99% of the atmosphere. Carbon dioxide constitutes only 0.04%. Atmospheric gas pressure is measured in millimeters of mercury. The air pressure at sea level is about 760 millimeters of mercury. The pressure of each individual gas is known as the partial pressure of that gas. The partial pressure of an individual gas is important because it determines the rate of diffusion between the alveolar air and the bloodstream. As long as alveolar oxygen partial pressure is higher than the oxygen partial pressure in the blood, oxygen diffuses from the alveoli into the blood. Each gas diffuses at a rate determined by its own partial pressure. The partial pressure of oxygen determines how much oxygen enters solution, but has no effect on the rate of nitrogen or carbon dioxide diffusion. Gas exchange occurs only by diffusion. No active transport mechanisms move respiratory gases in or out of cells. The direction that gases move is determined by the pressure gradients between blood and inhaled air. Even when this man exhales forcefully, some air remains in the lungs. Each new breath mixes with this deoxygenated air and decreases the partial pressure of oxygen in the lungs.
As a result, though the partial pressure of oxygen of the atmosphere is around 159 millimeters of mercury, the partial pressure of oxygen of alveolar air is only about 100 millimeters of mercury.
But the oxygen partial pressure of deoxygenated blood entering the pulmonary capillaries is much lower, only 40 millimeters of mercury. Oxygen, which follows the partial pressure gradient, diffuses from the alveoli into the deoxygenated blood until equilibrium is reached. So the oxygenated blood leaving the lungs has an oxygen partial pressure of 100 millimeters of mercury. A similar exchange takes place between the capillaries and the body cells. Cells constantly consume oxygen as they metabolize glucose in the process of cellular respiration. Diffusion of oxygen out of the blood and into the tissues occurs because the partial pressure of oxygen in tissue fluids is low, about 40 millimeters of mercury. Oxygen doesn't dissolve easily in blood plasma, and the amount that's transported this way isn't sufficient enough to meet the demands of large active organisms. As a result, many animals, including humans, have carrier molecules that increase the ability of the blood to transport oxygen.
In humans, this carrier molecule is hemoglobin. 98.5% of the oxygen in the bloodstream
is transported by hemoglobin molecules inside our red blood cells. The ability of hemoglobin to attract or release oxygen is primarily controlled by the partial pressure of oxygen in the local environment. These hemoglobin saturation curves show that as oxygen concentration rises, the amount of hemoglobin combined with oxygen increases. If the partial pressure of oxygen in plasma is high, hemoglobin is nearly 100% saturated with oxygen. The amount of oxygen carried by the hemoglobin molecules is called the percent saturation of hemoglobin. The percent saturation is highest in the pulmonary capillaries, where concentration of oxygen is highest, about 100 millimeters of mercury. The percent saturation is lowest in the tissue capillaries, where the partial pressure of oxygen is 40 millimeters of mercury or lower. The partial pressure of carbon dioxide in tissue fluid is about 45 millimeters of mercury. Diffusion removes carbon dioxide from the interstitial fluids from the drive of the partial pressure gradient. Blood entering the lungs through pulmonary capillaries has a carbon dioxide partial pressure of 45 millimeters of mercury. The carbon dioxide partial pressure of alveolar air is only 40 millimeters of mercury. So carbon dioxide diffuses out of the blood and into the alveoli. Carbon dioxide is transported from the tissues to the lungs using three methods. About 23% of carbon dioxide is carried by hemoglobin molecules inside red blood cells, and about 7% is dissolved in the plasma. The remainder is converted to carbonic acid, which breaks down into a hydrogen ion and a bicarbonate ion. So the next time you're jogging and you feel yourself breathing harder and harder, you'll know the type of travel it takes for air molecules to enter your lungs. Hi, I'm Susan Savoy, and welcome to Prentice Hall's video tutor discussion of kidney function. Throwing away trash is the most common way humans produce waste materials, but many times our garbage has valuable raw materials that can be recycled. Take these bins, for example. One is designated for recyclable material. The other is for non-reusable waste. The same decisions take place in your body. Your kidneys filter blood, regulating the chemical composition of your fluids. Some molecules are kept and recycled. Others are eliminated as waste. Nitrogenous wastes are produced as your cells break down proteins and nucleic acids. The most common nitrogenous wastes are urea and uric acid. Kidneys also regulate water and salt balance through a process called osmoregulation. So in order to understand these processes, we need to first take a closer look at kidney structure. Inside each of our bodies we have 2 kidneys. In each kidney we have millions of microscopic structures called nephrons. Each nephron is divided into 2 functional units, the renal corpuscle and the renal tubule. The renal corpuscle also has 2 parts, a cup-like chamber called Bowman's capsule and a spherical cluster of capillaries called the glomerulus that projects into the center of the capsule. From the Bowman's capsule, fluid enters the 3 sections of the renal tubule. The first section is connected to the Bowman's capsule and is called the proximal convoluted tubule. The proximal convoluted tubule leads to the loop of Henle. From the loop of Henle, fluid enters the distal convoluted tubule. Several DCTs empty into each collecting duct via short collecting tubules. The collecting ducts merge into passageways that drain urine from the kidney. Each nephron has its own blood supply. Blood arrives through a small artery called the afferent arteriole. The afferent arteriole supplies blood to the capillaries of the glomerulus. Blood is drained from the glomerular capillaries by an efferent arteriole. The efferent arteriole then delivers blood to the peritubular capillaries that surround the proximal and distal convoluted tubules and the vasa recta that parallels the loop of Henle. Blood flows from the peritubular capillaries into a series of larger veins until it leaves the kidney in the renal vein. Now that you're familiar with kidney structure, let's use this information to understand how urine is produced. Just as recyclable materials are further subdivided into glass, paper, and aluminum, our kidneys are involved in 3 main steps of urine production: filtration, reabsorption, and secretion. To get a better understanding of each step, let's go back to our diagram of kidney structure. Urine formation starts as fluid, and small molecules are forced out of the glomerular capillaries into Bowman's capsule. This process is known as filtration. Blood enters the nephron through the afferent arteriole, flows into the glomerular capillaries, and is drained from the glomerulus by the efferent arteriole. The narrow diameter of the efferent arteriole forms a bottleneck and elevates the blood pressure in the glomerular capillaries. The hydrostatic pressure in the glomerular capillaries forces fluid across a filtration membrane formed by the fenestrated endothelial cells, the lamina densa, and processes of specialized epithelial cells called podocytes.